Welcome to part 4 of this video series on the muscular system. This is the second video in which we will explore exactly how the nervous system stimulates skeletal muscles to contract. By the end of this video, you should be able to describe the resting membrane potential and explain why membrane potentials are important to muscle contraction. You should also be able to describe the propagation of action potentials in muscle cells. So, all cells have some type of resting membrane potential, but what is it? That's the question I had when I read about this topic for the first few times. I remember reading about this and crossing my fingers in hopes that it didn't show up on an exam. Hopefully, by the end of this video, you'll have a better understanding of the topic than I did when I was sitting where you are. Why should you not ignore this topic and just cross your fingers like me? Well, changes in resting membrane potential is the key to why neurons are able to transmit messages over distances of about a yard in the body, and why muscles can contract to move substances in our body or to move our bodies in space. So again, I ask, what is it? If we think about any cell in the body, it's surrounded by a plasma membrane. On the outside of that membrane, that's this side, we have extracellular fluid. The inner layer of the membrane, that's on this side, faces the cytosol, or the intracellular fluid. Suspended in both of these fluids are a number of ions, nutrients, nucleic acids, proteins, etc. For the purposes of this topic, we will focus on two ions that are most important for muscle contraction. They are your sodium ions and your potassium ions. Now, if you look at the distribution of ions on both sides of the membrane, you can see that there are significant differences. On the extracellular fluid side, that's the blue side here on the left, there are lots of sodium ions, which are represented by blue pentagons. The intracellular fluid, which is pink and on the right, has fewer blue pentagons, or sodium ions. Potassium, which is represented by the purple squares, is in higher concentration in the intracellular fluid on the right in comparison to the extracellular fluid on the left. Now, we can actually measure the concentration of both of these ions and kind of quantify how much there are. So what I've done here is on the side where you have the blue pentagons, the sodium ions, I've made the number for sodium really big. So 145 millimolar sodium concentration on the extracellular side. Compare that to 20 millimolar sodium ions on the intracellular side. So it's a smaller number, it gets a smaller font. The reverse is true for potassium. Potassium, we have 150 millimolar concentration on the intracellular side, so it gets a big number, and only 4 millimolar on the extracellular side, so it gets a small number. Now, what's not kind of included in this picture is this. The A here, the orange square, it represents protein. So there's a lot of proteins that are inside of the cell and in the cytoplasm, in the cytosol, but it's not in the extracellular fluid. So if you look at this diagram, you don't really see any orange squares or rectangles on the extracellular fluid side of the diagram. That's because they're basically made by the cell, and most of the time they stay in the cell. They can't cross the plasma membrane. These proteins exert a negative charge, okay, in comparison to the positive charges that we see with potassium and sodium. I didn't really talk about chloride in here because it's a minor component, and I want us to focus on kind of the most important ions here. Now, if the cell membrane was only a phospholipid bilayer, the difference in the concentration of ions would basically stay the same forever, and these cells that have these resting membrane potentials would be inactive. They couldn't do anything, really. Okay. But the plasma membrane is not just a pure phospholipid bilayer. Instead, we have proteins, like this one represented in purple here, okay, that allow certain ions to cross the plasma membrane. So this particular protein is represented in purple because it is a leakage channel for potassium. So potassium, that's the purple square, can move through this protein and cross the plasma membrane. So this is the figure out of your textbook that kind of explains what you saw on the last slide. So here's the plasma membrane, that phospholipid bilayer, and these are channel proteins that are embedded in the membrane. Now these proteins are called leakage channels. They basically make a tunnel through the plasma membrane that ions can move through, okay, to get from the outside of the cell to the inside and from the inside to the outside. Now these are specifically designed to allow the movement of potassium. And potassium is going to move through these channel proteins down its concentration gradient. So remember, that basically means that potassium is moving from an area of high potassium concentration to an area of low potassium concentration. Now, remember that on the last slide I showed you that the concentration of potassium inside the cell is something like 150 millimolar, and then outside it's only 4 millimolar. So that means based upon concentration, potassium ions have a tendency to move out of the cell and go into the extracellular fluid because there's a lot more potassium inside on this side of the membrane versus on this side of the membrane. Okay. Now, these are ions. That means they have a charge. Potassium has a positive charge, and if you'll remember from chemistry, positive charges are attracted to negative charges. Because of the proteins that are on this side of the membrane, on the intracellular side of the membrane, remember those proteins exert a negative charge, and because they're trapped on the intracellular side of the cell, of the membrane, that means that there's a strong negative charge inside the cell. So these positively charged potassium ions have a tendency to flow back into the cell, okay, based upon a charge gradient. So they're going to move away from positive charges back towards negative charges. So what this means is that potassium is going to flow out of the cell down its concentration gradient and flow into the cell down its electrical gradient. Okay. Now, if we were to stick an electrical probe into this membrane, let me see if I can draw one here, into the membrane. So this is my probe, this kind of triangle thing here. Okay, and we were to measure the change in electrical charge as potassium diffuses out of the cell and then diffuses back in, we would get a measurement of minus 90 millivolts. This is the resting membrane potential for your muscle cell. So voltage just is a measurement of the electrical potential across the membrane. And the electrical potential comes from the movement, really the separation of ions across the membrane. And it changes as ions move across. For us to get 90 millivolts, we have to have the same rate of potassium leaving the cell as is entering the cell. Okay. This is great and everything, but we didn't talk about sodium. Well, in addition to the leakage channels that are always open, so that means potassium can freely flow down its concentration gradient and down its electrical gradient, Okay. In addition to those channels, we also have gated channels that control the flow of ions. So while there are lots of leakage channels for potassium, there are also gated channels. And we've talked about those, right? These are proteins that have one or two closures, aka gates on them, restricting the flow of ions. So your sodium channels tend to be gated. And so that means that sodium doesn't flow as easily across the membrane. In addition to this, the membrane also contains what's called the sodium-potassium pump. So the sodium-potassium pump uses ATP to pump sodium out of the cell and to pump potassium into the cell. This is going to help maintain the distribution of sodium and potassium so that we have more potassium inside the cell and more sodium outside. The resting membrane potential is a measure of the separation of charges across the plasma membrane of the cell. Because of those leakage channels and the gated channels and the sodium-potassium pump, our concentration of sodium and potassium is always going to be different on both sides of the membrane. And this is going to translate to a difference in charge. Remember, we also have the proteins that are only on one side of the membrane that exert a negative charge as well. So that all of this means that charges on both sides of the membrane are going to be different. We say that the membrane is polarized because of this. And this polarization is a form of electrical potential energy. It's measured in millivolts. So what's important to remember about the resting membrane potential is that the interior of the membrane, that's the side that's facing the cytoplasm, it's going to have a concentration of negative charges because there's very little sodium on this side, and we have some anions like these proteins. Remember, anions are negatively charged ions, so your proteins kind of fit that bill. Okay, so the inside of the membrane has a net negative charge. Outside of the membrane, we're going to find a net positive charge. And this is because we have lots of sodium over here, very little potassium, and then we're missing those proteins. And if you look here, you'll see there's also a lot of chloride ions, but they contribute minor charges. Okay, so they're not going to really factor into this very much. So we have a very strong positive charge outside of the cell near the membrane and a very strong negative charge inside the cell near the membrane. And again, if we were to drop an electrical probe into the membrane and measure the difference in charge across the membrane, we would find that the resting membrane potential is about minus 90 millivolts for skeletal muscle cells. The minus here on minus 90 tells us that the inside of the membrane has a net negative charge. That's what you need to remember. Separation of charges, minus 90, inside of the membrane has a net negative charge. So you might be thinking, okay, Professor, I heard what you said, but I don't really understand why this is important. Well, first let's use the information that we just learned to draw in charges at the sarcolemma. So remember, the sarcolemma is the plasma membrane of the muscle cells. So as we learned just a few minutes ago, there is an accumulation of negative charges inside of the cell along the plasma membrane, and there's an accumulation of positive charges outside of the cell along the plasma membrane. We also talked about two ions, sodium and potassium, which are most important in the topic that we're dealing with here. So let's draw those in too. Remember that there's a greater concentration of sodium outside of the cell and a greater concentration of potassium inside of the cell. Okay, let's pick up from the last video. In the last video, I explained that an action potential travels down the axon of your motor neurons. So it comes from this direction. The action potential stimulates the release of acetylcholine, that's the green spheres here, Okay, it stimulates the release of acetylcholine from the axon terminus. Those acetylcholine molecules diffuse across the synaptic cleft, so this space here between the axon terminus and the membrane, that's the synaptic cleft. Acetylcholine travels across the synaptic cleft and then it binds specific sites on this protein in the sarcolemma of the muscle cell. So this protein is called the acetylcholine receptor, and it's really a ligand-gated channel. So remember, ligand-gated channels are channels in which a molecule binds them and causes them to open up or to close. So in this case, the channel opens up. This allows sodium to flow down its concentration gradient and potassium to also flow down its concentration gradient. So for sodium, that means that sodium is going to flow into the cell because it's going from an area of greater concentration outside of the cell to a lesser concentration inside. Potassium flows in the opposite direction because there's more potassium inside the cell, so it's going to flow out into the synaptic cleft. Okay, big deal. Why should I care about this? Here's why you should care about this. As sodium flows into the cell, the absence of sodium here at the synaptic cleft means that we're going to get an accumulation of negative charges. Now, because sodium has come into the cell, we're going to get an accumulation of positive charges inside. So basically what's happened is the polarity of the charges, the polarity of the membrane has reversed, okay? Because now negative charges are outside and positive charges are inside. That's the opposite of what we have in a resting membrane potential. This event is called a depolarization. A depolarization. So what's basically happening here, if we look at charges in particular, is when we have a resting membrane potential, so this is before acetylcholine has been released from the axon terminus, if we measure the resting membrane potential of this muscle cell, it's minus 90 millivolts. When acetylcholine binds the acetylcholine receptor, causing it to open up, and sodium starts to flow into the cell, this membrane potential begins to become more positive. That's what we're seeing here. Okay? If the membrane becomes positive enough, if the membrane potential becomes positive enough, that means enough sodium comes into the cell, we are going to get an action potential. But how does that happen? So when acetylcholine binds its receptor over here and it makes the membrane polarity reverse so that the inside of the membrane becomes more positive and the outside becomes more negative, that it corresponds to a change in voltage across the membrane. And we saw that on the graph, right? We saw that the resting membrane potential was at minus 90, and then when acetylcholine binds its receptors and the membrane becomes more positive, the membrane potential went up to minus 65. Well, in an adjacent part of the membrane, we have another type of protein channel, and this is called a voltage-gated channel. It opens or closes based upon changes in membrane potential. So if we get to that minus 65, again, that's called threshold, okay, threshold. If we get to that minus 65 millivolt number, because enough sodium is coming into the cell, then that triggers channels, which are called voltage-gated sodium channels, okay? It causes them to open up. Now, once these voltage-gated sodium channels are open, that means that sodium can flow across the membrane down its concentration gradient. So you know what that means. Sodium is going to flow from outside of the cell to inside. This makes this part of the membrane a bit more positive. Now, adjacent to the voltage-gated sodium channels, we have voltage-gated potassium channels, and reaching threshold causes these channels to actually close. So we're going to trap potassium inside the cell, that's a positive charge, and we're going to let sodium into the cell, which is another positive charge. So at this point, the membrane is becoming very positively charged. And adjacent to these voltage gated channels, there'll be more voltage gated channels. So what we get is something like the wave, where part of the membrane becomes more positive here, and then that triggers this part of the membrane to become more positive, and then that triggers this part of the membrane to become more positive, and so on and so forth down the muscle cell. So we talked about how when we reverse the voltage and the membrane becomes more positive, that's called a depolarization, because the difference in charge across the membrane is reduced because things are becoming more positive. So when these sodium channels, these voltage gated sodium channels open up, what we see is a change in that membrane potential. Here's where we started. This is when the acetylcholine receptors opened up, and now, because of these voltage-gated sodium and potassium channels, we see a large spike in positive charge. That membrane potential becomes much more positive, and it will go, this positive membrane potential will go all the way up to plus 35 or 35 millivolts. Okay. I think I said that a little bit strangely. Let me try one more time. So when the voltage-gated sodium and potassium channels open, the membrane potential becomes much more positive, and at its peak, it will reach about 35 millivolts in amplitude. Now, this is going to happen again like the wave. So one part of the membrane becomes positive, and then an adjacent part becomes positive, then an adjacent part to that becomes more positive, so on and so forth till we get down the cell. So this little animation here is supposed to represent that. The red bar shows you the opening of those sodium voltage-gated channels and the closing of the potassium voltage-gated channels happening at different sections of the membrane along the muscle cell. So depolarization of the sarcolemma is going to stimulate cells to contract. If we hope to contract the muscle cell again, however, after this event, we've got to return it back to its resting state. That means that the sodium that has been flowing into the cell, it's got to be pumped back out. The potassium that has been flowing, that's stuck inside, well, we've got to get it back to its proper concentration. We need to have that concentration of negative charges on the inside of the membrane and positive charges on the outside of the membrane. Repolarization is how we're going to achieve that. So if you remember on the last slide, we had a change in membrane potential that triggered these voltage-gated sodium channels to open up, and sodium flowed into the cell. Well, shortly after those channels open up, they snap closed. And so that prevents the influx of more sodium ions. Remember on the last slide, we said that the potassium channels, so these are also voltage gated, with the change in voltage, they start to close, and that traps potassium inside the cell. Well, during repolarization, those voltage-gated potassium channels begin to open up again. This allows potassium to start to flow out of the cell. So potassium will flow out of the cell through those leakage channels that we saw, as well as these voltage-gated potassium channels. So that means that potassium is actually going to start to flow out pretty quickly. Well, how do we get rid of the sodium that flowed into the cell? Right? We've closed the channels, but there's still a high concentration of sodium in the cell. Well, there is a protein called the sodium potassium pump, and it's an ATP ACE, so that means that it's going to use ATP to actively transport sodium out of the cell and transport potassium in. So what this protein does is it binds 3 sodium ions from the inside of the myofiber, the muscle cell. It binds 3 sodiums, breaks down or hydrolyzes ATP, and then tosses those outside of the cell. So we're removing 3 sodiums. Then it binds 2 potassiums from outside of the cell and brings them back in. So we're going to slowly adjust the charges across the membrane by stopping sodium from coming in and allowing potassium to flow out. And then we've got the sodium-potassium pump working as well. So what happens, if we look at this table, is that we were up here. Okay, and actually let me get rid of this stuff so that we can see. So we were up here on the last slide with the closing of the voltage-gated sodium channel and the opening of the voltage-gated potassium channel and the activity of the sodium-potassium pump. The number of positive charges inside the cell across the membrane begins to go down. And we'll see that the membrane potential becomes more negative. This means that our charges are separating again. We'll actually have a little bit of time at which the membrane potential will actually overshoot the resting membrane potential here because there's so much potassium that's flowing out of the cell. But the sodium-potassium pump will actually restore the concentration of sodium so that we will gradually reapproach that minus 90 millivolts, which corresponds to the resting membrane potential. Once this has happened, we can re-stimulate the cell to contract. So the events that I've described, where we had a depolarization of the membrane and then a repolarization of the membrane, that is, the membrane potential became very positive and then returned to being very negative, that is called an action potential. And those events happen here at the sarcolemma. Now you might remember from a couple of lectures ago, I told you that the sarcolemma actually goes down into the cell via these structures called T-tubules. So as action potentials are propagated along the sarcolemma, they will actually travel down the T-tubules into the cell. Now, as that action potential moves through the cell, it's going to stimulate voltage-gated channels that are here in the terminal cisterna of the sarcoplasmic reticulum. It's going to trigger those to open up, and this is going to release calcium into the cytoplasm of this muscle cell. And from that calcium, we're going to actually cause a contraction. But that's a topic for the next video. In this video, I've described the resting membrane potential. Remember, this is a form of electrical potential energy that is formed because of the unequal distribution of ions like sodium and potassium across the plasma membrane of a muscle cell at rest. Acetylcholine binding to receptors in the sarcolemma of a muscle cell trigger a change in membrane potential called an action potential. Action potentials are propagated along the sarcolemma, kind of like a wave at a football or a baseball game. They travel from one part of the sarcolemma to another part, and then to another and another, so on down the cell. As these action potentials travel down T-tubules, they trigger the release of calcium from the sarcoplasmic reticulum, and that causes a contraction. You must understand the topics in this video to do well on your next exam. Please reach out to me if you have any questions. I hope this video has helped your understanding of how action potentials are generated in muscle cells. I'll see you at the next video where we'll talk about contraction.